Hello, my name is Gwen McMillin, and I'm one of the medical directors at ARUP Laboratories over toxicology and pharmacogenomics. I'm also a professor in the Department of Pathology at the University of Utah. And today I'm going to present some of our perspectives on what baby would say if baby could talk, specifically about prenatal drug exposure and laboratory detection thereof. The Substance Abuse and Mental Health Services Administration here in the United States conducts surveys about national drug use patterns and trends. In the most recent survey from 2012, they reported that approximately 6% of pregnant women admitted to using illicit drugs during pregnancy. On this slide you can see the breakdown by age group, suggesting that teenage pregnancies are most likely to be exposed to drugs, whereas older pregnant women are less likely to use illicit drugs during pregnancy. Most Americans also recognize that prescription drug abuse has become of epidemic proportions, and this is likely to affect all ages as well. Florida is one state that has released data about the incidence of neonatal abstinence syndrome as a consequence of maternal opioid abuse. And this was reported in the CDC's Morbidity and Mortality Weekly Report back in March of 2015, indicating that they have experienced a greater than tenfold increase in the incidence of neonatal abstinence syndrome, or NAS, and that 99.6% of these newborns tested positive for opioid exposure. 97% of those were admitted to intensive care units, and the average stay was 26 days. Now, as less than 1% of these exposed newborns were documented to have been exposed to heroin, it's assumed that prescription opioid abuse is to blame for this. Overall, the Substance Abuse and Mental Health Services Administration estimates that 10 to 11% of births in the United States are affected by prenatal drug exposure. So what does this mean for the newborn? Premature birth and low birth weight are common consequences of in utero drug exposure. Withdrawal symptoms, and in particular the neonatal abstinence syndrome, which can be quite debilitating for weeks to months, is also observed. Later, there are feeding issues, failure to thrive, developmental delays, and behavioral problems observed with drug-exposed children. And drug-exposed children are likely to experience neglect and an increased risk of drug abuse later in life. So I think few would argue that we should ignore these children. We need to find ways to detect drug-exposed infants. So how do we do that? Well, the obvious way would be to ask the mothers if they've used drugs during pregnancy, but maternal self-report has been proven by many to be unreliable and to underestimate drug use. I've seen some situations where pregnant women are tested on a weekly basis for drug exposure, but this type of serial testing is not practical or cost-effective for most pregnancies, and some states would argue that maternal testing does not prove that baby was actually exposed. So the more common way to detect prenatal drug exposure is to develop some sort of questionnaire or algorithm to evaluate risk of exposure, either during the pregnancy, prenatal care visits, or at the time of labor and delivery. And then, of course, to test the newborn. So this slide illustrates a reasonable approach, wherein you might first consult local and/or state policies that are designed to dictate testing, reporting, and special programs that are available in your local area. Then use that information to develop a way to evaluate risk of drug exposure during pregnancy. Collect specimens and either submit those specimens for testing right away or hold those specimens to monitor the newborn for signs or symptoms of neonatal abstinence syndrome or other reasons for testing, and then submit the specimens at that time. So what specimens to test? Most people are familiar with the use of blood and urine for drug testing. However, these specimens are limited by the time frame or window of detection. That is, most urine and blood tests reflect only a few hours to weeks of drug use. When looking at a newborn and potential exposures during pregnancy, specimens like meconium and umbilical cord are preferred because they represent a chronic accumulation of drugs over the last part of the pregnancy. And I'll talk about those briefly now. So meconium is the first stool of the newborn, and it has been the traditional specimen for detecting drug-exposed newborns, with some 25 years of experience behind this specimen type. Meconium begins to develop as the gastrointestinal tract forms, somewhere around 12 to 16 weeks of gestation, and then accumulates along with drugs that the mother may have taken during pregnancy over the remainder of that pregnancy. Now, because the baby grows most during the latter part of pregnancy, the largest quantity of meconium accumulates during that time as well. Sometimes the meconium will pass during birth, but usually it passes at 48 hours or so after birth. Sometimes it takes even longer, particularly in drug-exposed infants. But it's relatively easy to collect by scraping off of diapers and looks quite different than a milk stool. However, nursing staff often complain about the coordinated efforts to collect and accumulate the meconium for drug testing, and a savvy mother may actually dispose of the diapers, preventing collection of that specimen type. So another specimen type that can be used for detecting drug-exposed newborns is the umbilical cord itself. So this tissue begins to form around the 5th week of gestation and grows with the fetus throughout the pregnancy. It's typically about 20 inches long with a full-term birth, so there's plenty of specimen to test. And it appears that drugs deposit fairly consistently across the length of the cord, so it doesn't matter what portion of the cord is tested. Umbilical cord is available from every child at the time of birth, which makes it very easy to collect. And for high-risk scenarios where time to result is critical, it may be the preferred specimen because it can be sent to the lab immediately after collection. Umbilical cord has sometimes been criticized because the concentration of drug analytes in the cord are much lower than those found in meconium. However, with appropriate technologies, it is possible to detect the drug analytes in the umbilical cord tissue, and these are examples of references wherein the specimen types have been found to be qualitatively similar. In our experience at ARUP Laboratories, detection rates are similar between the two specimen types. This slide illustrates positivity rates for 9 of the traditionally evaluated groups of drugs, showing that cannabinoids and opioids are the most frequently detected drug classes. So you've now requested some testing and get results back. What do you do next? This slide illustrates an algorithm for how to evaluate results. As with other clinical applications of drug testing, the results should be evaluated based on pre-test expectations. So the first question is, are the results consistent with expectations? If the answer is yes, then you proceed as per the relevant local or state policies for your particular area. If the results are not consistent with expectations, then it's a good idea to discuss results with the mother to find out if there are pieces missing from the history. It's also a good idea to talk to the laboratory to discuss limitations in testing or possible explanations, such as detection of drug metabolites or impurities in pharmaceutical products. Another important thing to investigate relative to unexpected positive results is the pharmacy history for both mother and newborn. And this is because both umbilical cord and meconium will detect drugs administered to the mother during labor and delivery. For meconium, sometimes drugs administered to the newborn are also detected, And this is important to consider relative to the time of specimen collection and the administration of drugs. So some frequently asked questions that we get. Can we detect first trimester exposures? And just based on the development of these specimen types in the newborn, it's extremely unlikely that first trimester exposures will be detected. Can we detect how frequently the mother used drugs, the time of last use, and or exactly how much was used? No. Concentrations generally do not correlate well with the pattern of drug use. Can we detect drugs administered in the hospital? As I mentioned, we definitely can detect drugs administered prior to specimen collection. That is, drugs administered to either the mother or the newborn. Can we determine how and when a newborn will be affected by drug exposure? No, the laboratory testing cannot do that either. What if results from twins or triplets don't agree? Well, this is actually a fairly common problem and is usually explained by either drug administration to one infant or not the other, relative to meconium anyway, the timing of drug administration relative to the specimen collection, or another major reason which would affect both meconium and umbilical cord is when the concentrations in one specimen are very close to the cutoff concentration and so tend to straddle positive or negative, whereas the other specimen does the opposite. And finally, what are typical patterns of results that are observed? So for this and some of these other questions, ARUP has conducted quite a bit of research, and these are examples of recent publications where we've explained the patterns that are observed in more detail. So to conclude, meconium and umbilical cord can both detect drug-exposed newborns. Qualitative results agree well between the specimen types for most drugs. The preferred specimen may depend on the hospital model for selection of newborns to test, knowing that umbilical cord is available right away and so could potentially produce results faster than meconium. Another advantage of umbilical cord is that it avoids detection of drugs administered directly to the newborn. Also, drug detection will reflect exposure based on test design, drug stability and recovery from the specimen matrix, drug use patterns, and timing of specimen collection. So it's important to consider all of those when you're interpreting results and to talk to the laboratory. And finally, you need to consider maternal admissions, pharmacy history for both the mother and the newborn, and consultation with the laboratory. So thank you for your attention.